Yes.
Yes. Okay. Hi, everyone. Thank you so much for coming. My name's Alex. I recently just defended this work as part of my PhD at Texas A&M University in Corpus Christi. The logo, it keeps messing up on me, but I work in the Coral Reef and Ocean Health Lab with Dr. Keisha Barr at the Heart Research Institute. And I looked at the metabolic responses of the genetically divided eastern oyster populations. This. That's not working. Sorry. Okay. So as a lot of you here may know, in Texas, we have a very unique eastern oyster population where the north part of the state and the south part of the state have different genetic makeups. So the pie graphs are showing us the northern genetic makeup in white, and the black genetic makeup is here in the southern part of the state, with this Corpus Christi Aransas Pass area acting as a transition zone. So this is zooming in on the transition zone. You can see denoted by the red and blue, the different genetic makeup and sort of how this transition zone is acting. Now, there are different hypotheses as to why we have these genetically different populations. It could be because of different geographical winds, tides, things like that, or it could be because of different environmental conditions that these two populations experience. So I was specifically interested in looking at the different environmental conditions, the local and global stressors that these populations are facing, to see if it explains maybe some differences in metabolic activity that they have. So as you also probably know, and I think you've seen this image already today, but the Texas coast is also very unique because of the salinity gradient, the dramatic salinity gradient that we see along the coast. So this is a difference in freshwater inflow gradient, where in the northern part of the state, there's high freshwater inflow, making the salinity very low. And in the southern part of the state, we have this hypersaline Laguna Madre estuary that is unique in the sense that really the eastern oyster populations in other part of the United States would not probably survive in these sort of conditions, but they do in Texas. So this salinity stress is what I'm considering to be the local stress. And then of course the global stress of climate change. We are seeing warming temperatures across the globe, but specifically here in Texas, and especially in the summer, we're seeing warming water temperatures, and the southern part of the state is seeing more tropical increasing water temperatures than the northern part of the state.
A non-print.
The northern, for my experiment, I had the northern and southern oysters that we tested, and these were all F1 generation from wild collected oysters on the Texas coast. So for the north and the south, there were 3 genetic crosses. These were all spawned at the AgriLife oyster hatcheries in Flower Bluff. They were strip spawned. And the differences in the genetic crosses is just where their parents were derived or collected from, and then the date that they were spawned. So the northern parents were from Galveston, Matagorda, Keller, or Oyster Lake, and the southern parents were from Upper Laguna Madre or the Brownsville Ship Channel. And over here, I just noted the different the average that these populations see. So in the north, the average salinity ranges from 15 to 35, and the average water temperature is around 26 degrees Celsius. And then you can see in the southern part of the state, the salinity is much higher, 45 to 50, and warmer at 32 degrees Celsius. So I used these water conditions to set up my experimental design. So, okay. My research question was, are there differences in the physiological response of these populations in Texas? So the way that I looked at this was by location, those north and south populations. I looked at metabolic response, metabolic oxygen and calcification rates, and then I compared the local and global stress of salinity and temperature. This is the experimental setup at the AgriLife oyster hatchery. You can see here one of the oysters in our intermittent flow respirometry chambers. And you can also see that there is a camera watching this oyster, and I'd be happy to talk more about the camera data some more. It's not included in this presentation, but it's an integral part of this study and it will be in the future publication. So the experimental design, I conducted 25 experiments from June 2023 to March 2024. And we had there were 6 chambers. Each chamber, one was the control, and then each treatment was tested individually and then interaction with the other stressor. So we had our low salinity and then low salinity and high temperature, control, high temperature, high salinity, and high temperature. For salinities, we have 10, 30, and 50. And for temperature, we use 25 and 32 degrees Celsius, again, based off the conditions that these oysters are experiencing. So this is sort of a mock-up of that intermittent flow design that you saw in the last photo. And the way that intermittent flow respirometry works is it's a method to measure oxygen consumption rates without the buildup of waste products such as ammonia or CO2. And the way that this works is that you have oxygen saturated water that is in your sump getting pumped up into your chamber. So you have a set period of time where you're pumping that water from the sump into the chamber. At a set period of time, that flush pump turns off. And now you have your measurement period here outlined by this brown box. And this is sort of like when you go to the doctor and you're going to get a stress test on your heart. You walk and then run, you walk and then run. And when you're running, you're under that period of stress. And when you're walking, you're not stressed. And they're measuring the change in your heart rate. So what we're doing here is we're measuring this oxygen consumption rate in the different treatments. After a set period of time, that flush pump is turned back on and the oxygen saturated water refills the chamber and you're ready to do cycle number 2. So you can do repeated measures over time without that buildup of waste product. So the oysters had 5 1.5-hour cycles. So one cycle of the intermittent flow was an hour and a half. And the 5 cycles in the full time series up there sort of looked like that. And you can see in the yellow below is the highlight of each of the oxygen consumption curves. And you can see that oxygen consumption curve 4 looks a lot different than oxygen consumption curve 5. Now, one of the good things about these repeated measures is you can sort of understand that variability, why these curves look different. However, it was also sort of a challenge of this experimental design because as we're hypothesizing and trying to prove from those oyster videos is that this was a time when the oyster decided not to open. If the oyster doesn't open, then it doesn't breathe. And as some of you might know, oysters can stay closed and hold their breath for very long periods of time. And that in some ways skewed our data. So this is a photo of the oyster in the chamber. And this is when the oyster is gaped, very wide and very happy breathing. That probably is what a curve would look like from number 5. Another measurement was the calcification rates. The way that we measured calcification rates of the oysters was using the alkalinity anomaly technique, where you can measure the total alkalinity of the chamber water and the total alkalinity of the sump water. And the change in this total alkalinity, you can extrapolate calcium carbonate formation based off the 2-to-1 ratio of a 2 molar change in total alkalinity is equal to a 1 molar change of calcium carbonate formation or dissolution. Okay, so what do we predict to see with temperature and salinity stress? We predict to see this sort of bell threshold curve where we have this optimal range when our stressor is low, and outside of our optimal range, we see metabolic suppression. So if we have metabolic oxygen on our y-axis, a larger negative number is going to be when the oyster is opening and happy and breathing. So they're breathing more so that consumption curve is higher, but consumption is negative. So, and then when we have our stress low and high salinity treatments, we expect to see that metabolic suppression. However, with the north and south populations, we expect the metabolic threshold curves to be slightly skewed, where maybe the southern populations would prefer the high salinity and the northern populations would prefer the low salinity. So let's get into some of our results. First, this is metabolic oxygen on our y-axis and the total weight of the oyster on the x-axis. And actually something that I want to do next also for the publication is to specifically look at the meat weight, not just the total weight of the oyster, to maybe that explain some of the metabolic response as well. But we did see a significant correlation where the smaller oysters were breathing more and the larger oysters were breathing less. So in this sense, this is similar to previous literature that has shown that smaller oysters can be more resilient to stressors. And we also see that at the oyster hatchery, where the smaller oysters can rebound and recover a little bit faster than the larger oysters. And when I say smaller and larger, I'm referring to like market size. So market size or sub-market size oysters. Okay, so now let's get into some of our treatments. We did not see any differences by population, season, or time point. So I put all of that data together here. So we just have our treatments on our x-axis, but I did still keep the north and south populations denoted by the blue and the red dots. Again, metabolic oxygen on our y, and the treatments are going from low salinity to low salinity, high temperature, the control and high temperature treatments here, and then our high salinity all the way to the right. And if you remember, this is sort of what we predicted to see with this optimal range of metabolic activity and the suppression in the sublethal range. And our data fits that trend where our control treatment, we're having the highest responsive metabolic activity followed by the high temperature. And we're seeing the most metabolic suppression in the low salinity treatments. So what does this mean when we look at the effect of temperature and salinity specifically, and we dive into that interaction a little bit more closely? We can see that there's an antagonistic effect where the primary driver of the decline in metabolic response is in salinity, and the temperature is slightly declining the metabolic response, but not as much as the salinity is driving it. We did also see a significant correlation between net calcification and metabolic response. So we saw that when the oysters were opening and breathing more, they had dissolution signal of their shell. And when they were breathing less, that's when we saw the calcification signal. So this is showing maybe some sort of metabolic trade-off happening between breathing and calcifying or growing their shell. However, the calcification data was very messy. There's a few reasons that this could be the case. One is that ammonia can affect total alkalinity, and salinity can also affect total alkalinity. We tried to account for the salinity, but we could not account for the ammonia. So we didn't see any significant differences or trends in net calcification. So in conclusion, we saw no differences in the thresholds between north and south. We saw metabolic trade-offs by treatment and size of the oysters. And we saw that salinity had a greater impact on metabolic suppression and interacted antagonistically with high temperature. So this data can be used to inform restoration suitability indexes for oyster restoration, and can also be used for selective breeding that's happening here in Texas to improve the Texas aquaculture industry. And future directions, we can look more into the dynamic energy budgets, look at that valve opening behavior with those videos, which is what I'm doing next for the publication. I think looking at clearance rates and also genetic variation in the lineages would be very important. And then we can ultimately provide that data for the suitability models. Thank you.
I think we've got time for one question. Anybody got one? Okay, good job.
Thanks.
All right. Next up, we have Riley Hawk. Okay. And the first talk title goes off the edge of the page, so I'm going to see it here. All right, she's going to be speaking about evaluating the extent of oyster mortality after a severe flood event in Galveston Bay.
Okay, awesome, thank you. All right, everyone, I'll make sure to talk nice and loud so everyone in the back can hear me. So today I'm going to be discussing the extent and severity of an oyster mortality event that happened last spring in Galveston Bay. So to just start with a little bit of background, eastern oysters can be found all along the Gulf Coast in our bays and estuaries, and they form both intertidal and subtidal reefs. However, I'm only going to be talking about our subtidal oyster reefs today. And although they can survive in a very wide range of temperatures, they do have those optimal ranges in temperature and salinity, which we just heard a little bit about. Previous research has found that greater survival is in temperatures of 20 to 26 degrees Celsius and salinities of 10 to 16 parts per thousand. And here in Texas, due to our warm waters and mild salinities, oysters in our bays can normally reach market size, which is about 3 inches in 1.5 to 2 years. So oysters are big business in Texas. Over the past 10 years, Texas has supplied approximately 21% of all US oyster landings. And then for Gulf Coast oysters, they represent about 30%. Galveston Bay, which is the largest estuary in Texas, makes up a majority of the oyster harvest. However, historically, the contribution of Galveston Bay was much higher. In the past 8 years, the average percentage of Texas oysters coming from Galveston Bay has been only 41% compared to 71% historically. And this is primarily due to a rapid decline in abundance of market-sized oysters, which really started in the early 2000s. This decrease can be attributed to a variety of different reasons, habitat destruction and habitat loss being one of the biggest factors, but also high harvest pressure, predation, disease, and climate change, which is causing severe droughts, flooding, and other natural disasters are another big factor. So the Galveston Bay team likes to liken oysters to Goldilocks. They don't like it too fresh, they don't like it too salty, they don't like it too cold, they don't like it too hot. High salinity paired with high temperatures increases mortality from predation, from oyster drills, diseases such as dermo, and then boring sponge, which eats away at the shell of oysters, is also more prevalent in high salinities. On the other hand, salinities lower than 5 parts per thousand decrease the growth and survival of oysters. In a low salinity environment, oysters close their shells and enter anaerobic dormancy, but they can only stay that way for so long. One study found that after about a week of salinities below 1 part per thousand, there was 100% oyster mortality. And at 5 parts per thousand with higher temperatures, mortality was greater than 50%. We estimate that Galveston Bay's baseline mortality rate is between 10 to 20%. So how do we assess mortality out in the field? So pictured here are a few oyster boxes or recently dead oysters with articulated shells or their shells still attached but with no tissue remaining. Studies on the East Coast have estimated that boxes stay articulated for 8 to 11 months. However, with higher salinities, that age or the number of months that those stay together may decrease. So studies in Texas would need to confirm that number. We can use the box count method to estimate mortality, which is the proportion of boxes to live oysters in our samples. So talking about this flood event from last year, a severe weather event on April 28th inundated the Galveston Bay watershed with areas north of Houston receiving up to 20 inches of rain. The main river feeding into Galveston Bay, which is the Trinity River, was above a major flood stage for 26 consecutive days. Shortly after this flood event, TPWD started receiving reports from oyster industry that there was widespread mortality. In response to this, both Chambers County and Galveston County issued disaster declarations in early June. So in response to these mortality reports, the Galveston Bay team conducted a specialized oyster mortality sampling in addition to our bi-monthly routine oyster sampling. So our routine oyster dredge sampling is part of our routine sampling program. We do 30 sample locations randomly around the bay each month. These sample locations include all known reef and is not restricted to only harvest areas. Data collected includes measurements of up to 19 live oysters, as well as counts of live oysters, brown shell, black shell, which is buried shell, and then spat, which are those baby oysters that have just recently attached. And then we also record the number of boxes in each sample. Our specialized oyster mortality sampling used the same technique, but with a couple key protocol differences. Our sampling locations were predetermined and included 2 to 5 locations per reef complex, which for Galveston Bay resulted in exactly 61 sampling sites. Locations targeted live consolidated reef, and 2/3 of our samples were taken on public harvest reefs. Data collection included only counts of live oysters and boxes. So since 2019, we have been collecting these box counts so that we can estimate mortality. So this graph here shows our average percent mortality per month since 2019. Previously, I stated that oysters prefer temperatures between 20 to 26 degrees Celsius and 10 to 16 parts per thousand for salinity. Unfortunately, our bay doesn't really meet those conditions very often, but there have been a few instances, and during those time periods, you can see that our average percent mortality is between that 10 to 20%, which is why we're kind of assuming that as our baseline mortality rate. So mortality in oysters is thought to occur after 2 to 3 weeks of exposure to salinities less than 5 parts per thousand. Since we started collecting this data in 2019, there have been 3 instances where average monthly salinity has been below 5 parts per thousand. In 2019, 2021, and 2024. For the low salinity events in 2019 and 2024, we can see a sharp increase in mortality. However, for 2021, we don't see that same response, and we are not really sure why. So going back to this flood event from last year, salinities before the flood were in that Goldilocks range of 10 to 16 parts per thousand from February through April. And we can see that we had low rates of mortality in those few months. After the heavy rain, salinities plummeted to 3 parts per thousand for the first half of May, dropping as low as 0 parts per thousand in Trinity Bay, which is where the mouth of the Trinity River leads to. Galveston Bay as a whole experienced average salinities below 5 parts per thousand from May 1st to July 1st. And as a result, our oyster mortality estimates were greater than 20% from June to August. As I mentioned previously, oysters can survive for a few weeks in those low salinity environments, which is why we don't see an immediate spike in May, but we see higher mortality the following month. And for our estimates, mortality peaked in the second half of June with an average percent mortality of 64%. So what areas of the bay were most affected by this flooding event? So there's a lot on this chart. Let me kind of walk you through it. So the light pink is our oyster footprints throughout the entirety of Galveston Bay. We have some private oyster reefs here, which are in a dark purple, restoration sites in tan, and data from our sampling. Oyster mortality sampling, which was our specialized sampling event, was in green, and our bi-monthly routine sampling is in yellow, and this includes June and July samples. The size of the circle represents the percent mortality, with larger circles having higher percent mortality. And then there are some diamonds on here, which are sampling locations where neither live oysters or boxes were collected, so no mortality was able to be calculated. And then there are also our harvest areas here. So the TX4, TX3, those areas, and that is how TPWD manages the harvest of our oysters during the oyster season. So now let's zoom in on TX1 in East Bay. So East Bay experienced lower rates of mortality compared to other areas of Galveston Bay. And this is primarily due to the lower influence of freshwater coming from the Trinity River. We found that for this area, the average salinity was slightly above the rest of the area, the rest of Galveston Bay, which is why we're assuming there was lower mortality here. All three of these reefs did experience higher mortality rates than the average, with the whole area experiencing a mortality rate of 19%. TX1 is a good representation of why this additional sampling was needed, as Middle Reef, which is in the middle here, was only sampled once for routine sampling and didn't provide any live oysters or boxes, so we weren't able to assess mortality. So the additional sampling here was able to provide that data for us. So now we're going to go up to TX4 and TX3, which is in Trinity Bay, which has the most influence from the Trinity River. Due to high discharge rates from the Trinity River, this area of the bay is usually fresher than the rest of Galveston Bay and experiences oyster mortality most frequently. Every sample that was taken during our specialized mortality sampling for these two areas resulted in a 100% mortality. Of the routine samples for TX3 and TX4, 6 of the 7 had no live oysters or boxes, so we didn't have any mortality data. But the one that did, again, had 100% mortality. This is unsurprising, as salinities were below 1 part per thousand for the Trinity Bay from May until mid-July. So now we're going to look in the middle of the bay here, which is kind of in between our freshwater inputs and our tidal inputs, which bring in the majority of salt water. So this area of the bay is the most productive for oysters, since it's in between and has those mild salinities that are best for oyster growth. This makes it important to gather as much information as we can from this reef, because it tells us a lot about our health as a whole. Due to our routine sampling program being based on a randomized sampling design, it is possible for a harvest area to not have any samples for a few months, which was the case for TX5 here at the top. Without any data for TX5, it was difficult to estimate how the reef complexes in that area were impacted by the flooding event, which is again why this additional mortality sample was needed. So during that mortality sampling, we estimated that the TX5 area of North Bar, which is the middle reef there, had about 37% average mortality, whereas the TX7 side experienced mortalities of around 44%. However, mortality estimates calculated from our yellow samples came to about 71%. So overall, oyster mortality for Galveston Bay due to this flood event was around 49%, although some areas were more affected than others, as we saw. TX3 and TX4 experienced 100%. The middle of Galveston Bay experienced 37 and 55%. And then finally, TX1 and TX6, which is on the west side of the bay, were still elevated at 19 and 17%, just less so. These lower rates are primarily due to distance from the river outputs, and then also the Houston Ship Channel that separates TX6 from the rest of the bay and influences the hydrological characteristics of that area. So when we compare data from the two sampling methods, we see that results were very similar. However, without this additional sampling, we would not have been able to estimate mortality in TX3 and TX5, which helps us when we're making management recommendations. If we want to look further and separate our data into the specific reef complexes, we again see that there are similar mortality estimates, although routine sampling did lead to slightly elevated levels of mortality, or estimated levels. The asterisks beside some of the reefs down here are reefs that have private harvest areas as part of them. And we can see that almost all of those areas have elevated levels of mortality, which backs up those claims from industry that there was widespread mortality due to this flooding event. So some conclusions. As a result of this mortality event, market CPUE dipped below last year's averages for the fall, and our pre-harvest season sampling conducted in September showed that there was very low levels of these market size oysters, and so they were not eligible for opening for public harvest, which is the first time that that's happened for Galveston Bay. I'm gonna skip ahead really quick because you guys can read. So this is important because as these events continue, more floods and more droughts, we're going to see these mortality events continue and possibly be more severe in the future. And so it's important for us to be able to conduct this additional sampling to fully grasp the extent and severity of these events so we can keep an eye on the health of our oyster reefs for Galveston Bay and the rest of the coast. All right, and I think that is all.
Really quick, we have some free seats up in the front if anybody is standing or afraid of us. If we don't bite, feel free to move up and take these seats. Thanks so much. We're gonna try to avoid falling behind here, but I'm sure she'd love to answer any questions. Our next speaker is Bailey Lynn.
Hi.
All right. So Bailey's talk is called Assessing Mesozooplankton Dynamics in Response to Two Different Seasons in a Texas Estuary.
Hi, I'm Bailey, and like he said, I'm going to be talking about mesozooplankton dynamics in response to two different seasons in 2023 and 2024 in Galveston Bay. And I am here to tell you that not everyone in this session is talking about oysters. So some quick background, estuaries are very vulnerable to a lot of different anthropogenic stressors and natural disasters, only increasing as a result of global climate change, including increased eutrophication, increasing bottom water hypoxia, invasions by invasive species, and increasing hurricane activity, which is especially relevant for us on the Texas coast. So because of their nature being highly sensitive to environmental change, as well as being poikilothermic and drifters, zooplankton are often used as indicator species for climate change. They're great at that. So Galveston Bay specifically has been subject to a lot of long-term human impacts and natural stressors over the past few decades, including oil spills and two major hurricanes, Ike in 2008 and Harvey in 2017. So developing zooplankton in the bay as an ecological indicator is very necessary for management of these Galveston Bay ecosystems, especially because zooplankton as indicators in our region in Galveston Bay are relatively understudied. So establishing a baseline for them as a community is really important. So since September of 2022, we've been sampling zooplankton at 5 different sites in the bay along the Galveston Bay line that was established in the wake of Hurricane Harvey in 2017. This line follows along a known salinity gradient in the bay and is also adjacent to the Houston shipping channel, although not directly in the shipping channel. It's about 2 kilometers off the shipping channel. So at stations 1 and 2, typically we see the most coastal influence from the Gulf of Mexico in the lower bay. Moving into station 3 in the middle bay, you see more of an interface between riverine influence and the coastal influence. And then at stations 4 and 5, you see the most influence from the San Jacinto and Trinity rivers. So moving on to this. So what do we do once a month when we go out? At each site, we collect environmental parameter data as well as the zooplankton data. So we'll take a surface water sample that is taken back to the lab and processed for AAA via fluorometry, and then also take temperature and salinity data with a YSI. For the big money moment, which is the plankton tow, we have 2 nets, a 100 micron net and a 200 micron net that are towed horizontally behind the boat, pulled back up on board, rinsed, preserved on board, and then taken back to the lab where I have the distinct pleasure of enumerating and sorting them to the lowest taxonomic level possible under the microscope. So my eyes are doing great. And typically the lowest taxonomic level possible in this case, especially for the copepods, is to the genus level. although occasionally there are some species level IDs too. And in total, we've collected we have collected 303 samples. And the data that I'll be focusing on today is taken from the 100-micron net specifically. So this is just a very brief look at the average baywide environmental patterns taken as an average in all stations across the time period from September 2022 to August of 2024. So you can very clearly mark out seasonal changes. Very clearly you can tell, oh, it's wintertime. Oh, now it's the summer, moving back into so and so and so. And you can see similar patterns in the salinities as well. And I would also like to point out specifically May to August of 2023, which were the months that in our case were the most affected by the drought in 2023. And then, as was previously mentioned, the freshwater inflow that we had in 2024 in the spring through summer, through the spring into early summer, specifically focusing on May to August as well and comparing these two seasons. So what do we see in terms of salinity? At station 1, typically in 2023, you would see very typical for the bay, between 20 and 30 salinity, most coastal influence at 1 and 2. And then moving forward, you see that transition into being more of a mix. So station 5, typically values are between 10 and 15. And that holds true for most of these months, with the exception of August 2023, when the heat was at its hottest. And salinities, for the most part, even up at station 5, were in the 15 to 20 range, which is relatively atypical. And then you see surface as well to the bottom. That's just noting that. And then you see in 2024 on the bottom at May and June, most specifically here, the lower salinities even at the mouth of the bay, then moving up into stations 4 and 5, where salinities were again between 0 and 5. And just anecdotally, I can say that during one of our sampling trips, I accidentally got a mouthful of water and it was quite fresh. So you can see then returning more to a state of normal, so to speak, for the bay in July and August, although salinities in comparison with July and August of 2023 were still lower. And pairwise comparisons of all of these months did show that there were significant differences in salinity between all of these months in this time period, May to August. So what does that mean for our community? What does the zooplankton community look like in the bay? So here we have station 1, 2, 3, 4, 5, and then May 23, May 24. So 23, 24 side by side. So for the most part in both years, the community is majority copepod and copepodnoplei. And the copepodnoplei are in the dark purple, and Acarsha, the calanoid species that is most dominant at all stations in the bay, is in the light purple on top. Generally also true in both years, you'll see Oithona, which is a smaller cyclopoid species as well. Although as you move up into the bay further, you see it less, as it is more of a higher salinity species, or genus in this case. So specifically, something I want to point out in 2023 versus 2024 is this light pink color, which is a copepod known as Paracalanus, which is also a calanoid and is known to prefer higher salinity environments and to have a lower tolerance range in terms of that. And you can see that actually in 2024, specifically as you move forward, it's absent or in much smaller numbers at station 3, whereas you can see in June of 2023 that it was still present. So 2023, you can see clearly very much so at the lower bay stations, just visually more diversity. And that holds true as you move up through to the upper bay as well. And then specifically in 2024, I encountered some species that I had not seen before, which threw me for a loop for a bit. And that is Mesocyclops, which is a freshwater copepod, which was likely pushed into the bay from the Trinity or San Jacinto River, which was only present in May and June at the middle to upper bay stations, as well as Ergasilus, which is a type of parasitic copepod that is known to be oligohaline, which I had also not previously encountered until the freshwater inflow event in 2024. So overall, that is what the community looks like. Oh, and additionally, there were also various Cladoceran species that were suddenly in the bay as well that typically were absent and in 2023. And then as things return to normal, they cease to be in the sample as well. So After an analysis of variance, I found that in 2023, within that year, there were significant differences in diversity by the station, specifically between the lower base stations and upper base stations, so GBL 1 and 5, GBL 2 and GBL 5. But there were no significant differences in density either monthly or by station. And then in 2024, there were actually significant differences in density by month, and that was between May and June of 2024. And there were additionally still significant differences in diversity by station with the same stations, but with the addition of stations 2 and 3, which typically are much more similar as, locationally, they're relatively close together. But station 2 during the freshwater inflow event was not as affected as station 3 was. So then after a MANOVA, I saw that there were significant differences in community composition between both summers. So generally, composition was affected by the differences in the environment. And then this is just a look at the biological parameters and the environmental parameters taken together. So in 2023 and in 2024, both years, there was a significant strong positive correlation between diversity and salinity. This is well established to be true in Galveston Bay, especially in the case of copepods. So typically where you see higher salinities, you're going to see more copepod diversity. And again, this holds true for both summers. But I would like to point out that in the summer of 2023, there's a significant correlation between diversity and temperature that is not present in 2024, which may imply that because of the drought and the increase in temperature that led to an increase in salinity, there was also an increase in diversity, whereas in 2024, that same thing did not happen. Where's the relationship between diversity and temperature? And then generally, you also see in 2024 an inverse relationship between chlorophyll A and density, which is interesting to know. It's weak, but it is significant. And it is possible that this is because of lag between chlorophyll A and zooplankton density, or due to species being advected into the bay at the mouth that are more coastal. So just general notes there. So overall, were there any significant differences within the two seasons? Yes, there were. There were significant differences in diversity across stations in both years. And there was a relationship between temperature and diversity that was significant in 2023, but not in 2024. And then between the two seasons, there were pairwise differences in salinity between all months, as well as differences in community composition between the two periods. And then just generally for future work, cruises along the Galveston Bay line will continue until May of 2025, which is next month, and hopefully beyond. And then this is just some fun times that we've had in the field. I just wanted to put up some pictures of field work, and I have a very brief video of us, or not me specifically on the boat, but just in the bay, driving around. Finally, I would like to acknowledge my funding sources. So thank you to TCEQ and GBEP, and then also to all of the wonderful people that have helped me. So my advisor, Dr. Hui Lu, my former undergrad assistant, Nathan Henderson, my lab mate, Jishwan Song, and all of these other wonderful people. There's too many names, not gonna list them, but yeah, thank you, and that's it.
It was a very nice talk that on second glance has nothing to do with oysters. Great job. We have time for a couple of questions. Anybody? Hey. One. Next speaker is Rebecca Muniz. You around? Hey. I think it's my sheet. Her talk entitled Microplastic Detection in the American Oyster and a Depuration Study collected on the southern Texas coast. It's fine.
Okay, good afternoon, everyone. I hope everyone can hear me. My name is Rebecca Muniz, and unfortunately, I will be talking about oysters. I am all over them, so let's just try to get this going. So I'm going to be talking about the microplastic detection, specifically in two locations. And I did a field and laboratory study, so I'm kind of like comparing the two to see what that means for our oysters and the microplastics. And so a little bit of background, again, I'm gonna go a little bit quickly and I should have cut down my slides, but I didn't, so here we are. So what is plastic? So plastic, as we know, is a polymeric material that can be molded and shaped by applying pressure and heat. It's very popular due to 4 reasons. It's very low in cost, production-wise or processing-wise. It's highly durable and resistant, like sunshine, moisture, rain, whatnot. Low in weight compared to like plastic or metals. And it's very versatile, as we know, it's basically in everything that we use, even in ourselves, so yeah. So not all plastic is the same. So there's like 7 main types of plastics, but because of time, I'm only gonna be talking about 3 of them. So the first one is polyethylene terephthalate. These are your jars, your food bottles, your rope, your polyester clothing, as we can see in the top picture. We have the polyvinyl chloride or your PVC. These are your credit cards, your plumbing pipes, your toys, and your medical tubing / bags. And it's kind of ironic because PVC tends to like leak really toxic chemicals, such as like lead. So that's fun. And then we have our polyester, or most popular known as styrofoam. These are your cups, your packaging, and your takeout food containers. So these are just some of the microplastics. Going on to the importance of microplastics, why should we even care about plastic in general? It is not biodegradable. That means that it will just continuously be getting smaller and smaller and smaller, but it'll never, again, biodegrade. So it's always going to be in our environment. It is found in every single body of water. So whether that be your oceans, your rivers, your lakes, and the ice, anywhere there's water, there's microplastics in there. Which means that it's very detrimental to everything on this planet. As we know, everything is connected. So yeah, it's a lose-lose for everyone. So a little bit of the plastic timeline. This is just from 1950 to 2022. But in 1862, there was the first example of man-made plastic. In 1907, the first synthetic plastic was created. 1950s, as we can see here, it starts. After World War II, plastic production increased. As we know, a lot of advances in the medical world and technology, sports, whatever. 1970s is when microplastics was first identified in the marine environments. And then 2004 is when microplastics was first used. So going into their size and classification, we have nano, micro, and macro. But as I said, we're only going to be focusing on the microplastics. So this is between 1 micrometer to 5 millimeters. And we have 2 types of microplastics. We have primary microplastics and we have secondary microplastics. So primary microplastics are the plastics that are already micro in size. So some examples you can see here are the resin pellets or the microbeads that we can like find in our cosmetics or like in our body scrubs and stuff like that. So these plastics are already micro in size as compared to the secondary microplastics. And so these are microplastics that come from larger pieces of plastic, so your macro, and then they eventually get smaller. So those are your secondaries. So primary, already micro, secondary, they get micro in size. So these are some of the main contributors. We have land flow, which is like textiles, like runoff, agriculture, stuff like that. Tourism, as we know, we like to go to our beaches. What do we take to our beaches? We take our chip bags and our plastic bottles and our Coke cans and whatnot. And what do? We leave them there. And so that eventually goes into our oceans. Transportation, these are like your cargo and fishing fleets. So they throw like their crates or their nets overboard. And then I would argue, and I don't like to argue, but I would say that fishing is the #1 contributor to plastic in the ocean. So again, as I said, everything is connected, right? It's a domino effect. So these marine organisms will eat these microplastics. It affects their health. And then we eat these marine organisms and that affects our health. As we can see here in the little graph picture. Oh, again, you can right here. So everything's kind of connected. These little ones will eat the microfibers or the microplastics. The larger ones will eat the little ones. And then they eventually end up on our dinner plates. And so these are just 3 examples of human effects of what oh Jesus. I'm so sorry. Oh, there we go. No, don't worry. So plastic has been known, such as polyvinyl chloride, to cause cancer, while phthalates are known as endocrine disruptors. And this is really recent in study, but there has been an increase in neurodegenerative diseases such as Alzheimer's. So we're finding microplastics in our brain, which is causing these diseases. And also very recently it's been found that it's been found in the human placenta or in the breast milk. And so my sister just had a baby and I think I got like too much in her head about it. And so she's kind of like crazy about plastic and like having plastic like around her baby. I mean, I feel bad, but then I don't also. But yeah, so there's different routes of exposure. We have inhalation, so we can breathe in. Breathing in is more like nanoplastic. Ingestion, again, nano and like micro, which is like what we get from our food. And then dermal, so it can go again more like nanoplastic. but we can still get dermal contact. Yeah. So these are just some fun statistics that I found. Okay. So every week we consume about like 5 grams, which is like a soup spoon. And then every month we consume 21 grams, and every 6 months, 50 grams, 125 grams, sorry. Every year, 250, every 10 years, 2.5 kilograms. In our lifetime, we will consume 2 recycle bins, which is so fun. And again, I don't know why they're coming out like this, but again, some microplastic statistics. About 44 microplastics per gram of nano were found in sugar. 0.11 were found in salt. 32.37 were found in beer. 325 were found in bottled water. And it's estimated intake of 80 grams per day of microplastics via plants. So I'm a vegan, so I'm still getting my daily dose of these microplastics. So no problem there. And I thought this was a little like fun representation that we can see. So the top is from 0 to 6, and this one and the bottom is more like zoomed in. It's just from 0 to 1.5, and we can see different types of food and the amount of microplastics. So again, I like to look at the very second to last, the plant-based food. Again, that's what I eat, so I like to still be represented in these. So now going into a little bit of my study objectives. So I had 3 study objectives, and the first one was to examine the presence of microplastics and the polluted site. So this is my field study. And then my second one was to determine the recovery of these microplastics under controlled laboratory conditions. So this is my deparation study. And then my third one is to determine the effects of microplastics on oxidative and nitrogen stress and the antioxidants in the oysters. So going into a little bit of the American oyster, it's found along the Gulf of Mexico. As we know, they are filter feeders, which means that they are very important ecologically. And then they're also seen as delicacies in a lot of places. So economically, they bring in a lot of money. So these are important little guys. So this is an oyster picture that I took, open from the shell. So we can see the shell is there. And then the gills are like the little black flaps that are there. We have the extrapaleo fluid or body fluid. We have the adductor muscle, which is what we have to squeeze to get the tissue. And then we have the digestive gland. So I only focused on the gills, the extrapaleo fluid, and the digestive gland. That's all I took from these oysters, which is really all there is to an oyster. So I had 2 study sites. So we had South Padre Island and San Martin Lake. And I got around 15 oysters in both sites. And as we can see, a lot of plastic and a lot of pollution there. So as I said, I had field and lab. So my field, what we did is we went to these 2 study sites. We got the oysters, we took them back into the lab, and immediately we sampled them. We got their tissue and their body fluid that same day. When I say deparation or lab, that means that we went into these fields, we got these oysters, we took them back into our wet lab, and they were there for 2 weeks under clean water. And I cleaned the tank and I siphoned the water every other day. And we checked their water temp, pH, and DO. So the lab, they were able to kind of a little bit of a recovery. I know it's 2 weeks isn't that much of a long time, but we gave them 2 weeks. And so we're trying to see the difference between lab and deparation. I mean, lab and field, sorry, or deparation, same thing. So this is my experimental design. As I said, about 15 oysters per site. Yeah. And so going into the actual tissue collection, they were preserved in 4% formaldehyde for about like a week. Then we removed the water using alcohol and xylene. And then they were embedded in the paraffin wax for further analysis to stain them and whatnot. Yeah, that's me. So this is my first type of staining. I did about 4 types of staining. So I did the now red staining first. And so this is a fluorescent stain. And this was specifically to detect and quantify the microplastics. I just wanted to see the microplastics and see how many microplastics were in the tissue. And so the hypothesis would obviously be that the field has more microplastics than the lab, because the lab, they were able to kind of have that recovery period for 2 weeks, right? And so here, yeah, you can't really see them, but the field on the top and the lab on the bottom, the field, they had larger pieces compared to the lab, as we can see in the results. Again, lab is deparation. So the size of microplastics, there was significant a decrease in them. But again, this is where it kind of becomes tricky, because we didn't test the water chemistry of the tanks. So I don't know if it's just like the larger microplastics becoming smaller, but it's still the same amount of microplastic, or if like they were actually like siphoning these microplastics out. I cry about it, so, but there's nothing we can really do. So this is the digestive glands, and then these are the gills. I will say the gills had more microfibers as opposed to like microplastics. Again, all microplastics, but these were more fibers. And these had a greater significance in size. So these were significantly smaller in the lab as opposed to the field, which would make sense. So my second type of staining was periodic acid shift staining, also known as path staining. And so this was to detect the mucin. So I wanted to see the mucus concentration of these, since we are filter feeders. And so when they get the water, they use that mucus to keep all of whatever the algae, or in this case, the microplastics. So we wanted to see how the mucus is being affected. And so these pictures are kind of not great, so please just ignore them. But look at the results. So obviously, the field, they're under more stress, so they wouldn't produce as much mucus, which we can see in my results. My field, their mucus secretion was significantly lower as opposed to the lab. In the lab, they had a more, common or more better mucus secretion. And then we can see that as well in the gills. It's not as significant in the gills, which is kind of ironic, because that's where the mucus is. But there was still a significant difference between field and deparation. So then again, that goes along with our hypothesis. Our third and final, I kind of just said three. Our third and final was immunohistochemistry analysis. So this was to target specific antigens or the proteins in the tissues using the targeted antibody. So we had that primary antibody, which will bind to the specific antigen. And then we have that secondary antibody, which will bind to the primary antibody. And that's what causes that change in color. And that's what we use to see the targeted protein. And so the first type that I did was NTP expression. So NTP is a biomarker of nitrosative stress, and that's just really bad for the cells. And so a higher NTP equals that they're under higher stress. So we don't want a high NTP expression, we want a low NTP expression. So the hypothesis would be that my field oysters would have higher NTP expression because they are under more of that stress, right? As opposed to my lab ones who were able to chill for 2 weeks. And as we can see right now, a darker brown is going to indicate that higher NTP expression. I'm still getting my data for this one, so I'm not going to show it. It's not very good. But we can visually see that the field has like significantly darker in color as opposed to the lab. So that's a good sign. That means that my field were a little bit more stressed than my lab, which is what we were trying to see. My gills is a little bit, the results aren't really significant, I would say, but it is lower in my deparation or in my laboratory, but we can see it better in the pictures. The field in this one is significantly darker, and then the lab is significantly lighter, which means that they are under more stress. And then I also did 2 types of antioxidants. So I did CAT and SOD. I have 2 minutes, so I'm only going to kind of brush through these. And so when they're under higher stress, that means that they are producing more of these antioxidants to kind of combat the oxidative stress. So I did SOD, which is my first one. And so my field would have higher antioxidant expression because they're under more stress. So they're trying to combat more of this stress. So that means that they would have higher antioxidant expression, right? Which we can see here. The IR intensity of my field is a little bit higher than my deparation. And again, you can see that in my gills as well. It's not as significant as the digestive gland, but you can still see that there is a little bit higher in my field than in my deparation, which makes sense. You can see a little bit clearer in my CAT. So I did SOD and CAT, which are my 2 types of antioxidants. This one was a little bit more significant. I don't know why, as opposed to the SOD. I'm not really sure why. But my field study did have a higher IR intensity, or SOD, the CAT expression, than my deparation. And the same can be said for my gills as well, as we can see here. Oh, yeah. Not as significant as my digestive gland, but it still goes along with our hypothesis that my field are working a little bit more to produce these antioxidants to combat that more stress that they're under. And so in conclusion, there are more microplastics detected in the field digestive glands and gills than in my lab, which was what we were trying to see. Oysters experience stress from microplastics altering their mucin production, as we saw with the path staining. We know that humans are consuming these microplastics when they're eating oysters raw, because we now saw that the oysters do have microplastics in them, which means that microplastics impair the health of oysters and humans, and therefore need for better waste management is crucial for a healthy ecosystem. And that's it. Oh, I don't remember putting this. I guess I was in my field last night. But these are some quotes. We are living on this planet as if we have another one to go to, and for most of history, blah, blah, blah. If you can read it there. That's it. Thank you so much.
Time for one quick question.
If you guys have questions, I also have questions. Yes.
And once they get the microplastics in them, are they stuck with them?
Well, that's what I'm saying. I didn't test the water chemistry, so I don't know, like, I wish we could have tested to see, like, before cleaning out the tank, how much microplastic was in the water, cause I don't know if the microplastic is just getting smaller in my field. It's the same, it's like the same microfiber, but it's just like broken down. I don't know. And like I said, I cry about it because we should have gotten the water chemistry and I didn't. And I won't do it. I won't go back, but. That's what we got.
Thank you so much.
Thank you, guys.
All right, we've got one more speaker for our first session. So our next speaker is Mohammed Amin. The title is Effects of Short-Term Exposure of Antidepressant Drugs on Tissue Architecture, Body Fluid Conditions, and Nitrated Stress in American Oysters.
Good afternoon, everyone. I am MD Faisal Amin. I'm an international graduate student, Master's in Biochemistry and Molecular Biology from the University of Texas, Rio Grande Valley. So today I'm gonna present my master's thesis regarding the effect of short-term exposure of antidepressant drug on tissue architecture, body fluid conditions, and the nitrative stress in American oysters. So my research actually focusing on the effects of the pharmaceuticals that are entering to our marine environment and how it is caused destruction and hamper to our marine organisms and ecosystems. Let's talk about pharmaceuticals. So as we know that pharmaceuticals are some chemical compounds that is used for the treatment of diseases, diagnoses, as well as to treat different conditions. Pharmaceuticals includes medications, vaccines, and biologics. So pharmaceuticals and drugs are normally used for the control of infectious disease, and pharmaceuticals are manufactured through an extensive research and testing. So now let's have a look. This is the diagram that is representing how pharmaceuticals are entering through multiple steps and finally mixing to our marine environment. So in this diagram, as we can see, so industry that is producing personal care products and pharmaceuticals, and it is going to the hospital and clinics and diagnostic centers, and we people are taking these medicines as well as the veterinary animals they are also taking. And after taking pharmaceuticals, we ingest pharmaceuticals and it is metabolized in our body, and the metabolites and the end products ultimately goes to the sewage system through the excretory system. So these end products and other wastewater treatment from the industry all are going to our marine environment and causing harm to our marine ecosystem and marine species. So that is the scenario that is representing how pharmaceuticals are causing harm to our marine ecosystem. So let's have a look about the impact of pharmaceuticals on marine organisms. I have said that pharmaceuticals have many detrimental and noxious effects like bioaccumulation, endocrine disruption, genetic mutation can be happen because of pharmaceuticals, as well as the behavioral changes, impaired reproductive functions, and ultimately it causes ecological imbalance. So that is a figure, is a fish formed by different dosage forms like capsules, tablets, etc. That means our marine organisms are now getting destroyed because of pharmaceuticals. Now let me talk about the specific drug that I used for my experiment, and it is the fluoxetine, very commonly used antidepressant drug, and young generations are taking this drug very much than any other drugs in order for the treatment of depression nowadays. So this drug fluoxetine is commonly named as Prozac, which is a commercial name and discovered by the very famous pharmaceutical company Eli Lilly, United States of America, and it got an approval by the Food and Drug Administration in 1987. And that is the chemical structure of fluoxetine at the right side of the slide. And so this chemical I used as a standard chemical for my experiment. So now let me talk about the study objectives that I had during my experiment. So my first objective was to examine how the antidepressant drug fluoxetine impacts on oysters tissues, as well as another objective was to assess the effects of fluoxetine on oxidative stress in oysters and to investigate the effects of fluoxetine on the body fluid conditions of oysters. So now let's have a look about the American oyster. Already you have known about it. So it is very common and native to the Atlantic and Gulf coasts of North America, and it lives in estuaries and coastal areas, and very important ecologically because it acts as a natural filter feeder, improves the quality of water by filtering the algae and other particles, and very important economically. So oyster is very common food, and if the oysters is getting polluted, and ultimately it's coming to our food menu, and a dish of oyster. So if the oyster is getting polluted, ultimately we are getting the harmful effects because of the chemicals. So the drug I used, fluoxetine, I took 2 different doses. One is low dose and another one is high dose. And for the control, there is no dose. So for low dose, I took 0.5 microgram per liter, and for high dose, I took 5 microgram per liter of fluoxetine. So here is the figure that is representing my experimental setup. I took 6 aquariums, but here I have shown 3 aquariums. One is for control, and another one is for low dose, and the third one is for high dose. And each tank contained 15 oysters, 15 or 16 oysters. So the reason why we took 2 specific organs for my experiment in oysters. Number 1 is digestive gland, very important for digestion in oyster, as well as the another important organ in oyster is gills, very important for the filtration of water. So these 2 specific organs I used for my experiment. So basically, I performed histological analysis, and here is a representation of the histological analysis method. The microtome machine we used to cut the paraffin block, and the ribbon of tissue section, as you can see, and the paraffin block I prepared during my histological experiment. So different software I use for collecting data and my statistical analysis, like ImageJ and GraphPad Prism, very popular and commonly used tools for statistical analysis, we know. So now let's talk about the result of low dose and high dose of fluoxetine on oyster's digestive gland. So the first picture is showing the control where no fluoxetine used. And as you can see, the very uniform and natural healthy look of the digestive gland in control. But in low dose and high dose, you can see, though it is not very easy to see by our eyes, but there is a effect in low dose, and in high dose, as you can see, the deleterious effect of fluoxetine is representing by the expanded lumen area of the digestive gland, and in low dose the same. And because of the effect of drug, the epithelial lining of the lumen has become smaller. So this is the whisker plot is representing the statistical result of the low dose and high dose. The red arrow sign is indicating the enhancement of the lumen area because of the effect of fluoxetine compared to the control. Now let me talk about the effect of fluoxetine on the oyster's gill. So in control, the oyster branchial filament is very uniform and parallel. But in low dose and high dose, you can see the destruction of the branchial filamental cell, right? And the intralamellar area in gills has been expanded, as well as the area of gill lamellae has been increased significantly because of the high dose compared to the control. So another staining I did during my experiment is periodic acid ship staining. So here I used periodic acid and ship reagent to determine the amount of mucus in the digestive gland and in the oyster's gills. So this is a container of ship reagent, and this is a slide of my experiment where I did my periodic acid ship staining. So let me talk about the result of the PAS staining. So in control, as you can see, these substances is the mucus in control, and you can see very uniform and very natural amount of mucus, but in low dose and high dose, the amount of mucus is decreasing significantly because of the effect of fluoxetine. That is representing that fluoxetine is is decreasing the amount of mucus in oyster's digestive gland. And the whisker plot is representing the statistical analysis of the result in low dose and high dose compared to the control. So the effects of fluoxetine in oyster gills, as we can see, the purple color here is indicated by the arrow sign is mucus. So in control, very uniform and good amount of mucus, but in low dose and high dose, there is a significant lack of mucus production because of fluoxetine in oyster gills. That is representing that that fluoxetine is also producing its deleterious effect to destroy the mucus production in oyster gills. So this is the result of the body fluid pH level because of the fluoxetine in oysters. So we saw the extrapallial fluid pH level significantly decreased because of the drug. That means the body fluid is becoming more acidic because of the effect of fluoxetine in oysters. So another part I did is immunohistochemical analysis. So immunohistochemical analysis is a laboratory technique where two different antibodies are used. Primary antibody is normally bind with the antigen, and secondary antibody, which is linked with a biomarker that binds to the primary antibody and produces a different color, a particular color, highlighting the target protein. So now let me talk about my result. So I basically determined the 3-nitrotyrosine protein expression that normally produced because of the effects of different substances like different pollutants, chemicals, heat stress, pharmaceuticals. So in control, there is no expression of nitrotyrosine protein, NTP expression in digestive gland, but in control, in low dose and high dose, as we can see, the dark area that is representing and highlighting the expression of NTP protein in oyster because of fluoxetine. And the whisker plot is indicating the significant increase of NTP expression in oyster's digestive gland. So now let me show the effect of fluoxetine in NTP expression for gills. So in control, there is no expression of NTP protein, but in low dose, it will be low dose. I'm sorry, it has written control, but it will be low dose, this picture, and this will be high dose. So the significant nitrotyrosine protein expression can be observed because of fluoxetine. So that was the overall result of my experiment, and ultimately we have understood that our marine environment is endangered and in threatening condition because of pharmaceuticals that are coming from the pharmaceutical companies and other medicine and chemicals producing company. So this is representing how pharmaceuticals is hampering our ecosystems. So that's all about my presentation. Thank you very much.
Anybody have any questions?
Okay. Okay, sir.
Okay. So here's the immune system. Are they more susceptible to dermal?
Sorry.
Oyster disease. I know you don't think it, but you would think it would be more susceptible to dermal if they had an compromised immune system. Would be something interesting to look at.
Yes.
Can you give us a little bit of context with your high and low doses? How do they relate to measurements that have been made in the environment? And how did you choose those doses?
Actually, before selecting the doses, I researched literature. So what amount and what kind of concentrations are available in different research paper has been detected by researching the marine water from different regions. So from a particular paper, I used as a reference for selecting this particular dose for low dose and for high dose.
Anyone else? Thanks so much. All right, we're all done in here. There's a poster session next that's in the Lyceum, the big long room with the windows. The sure you went through at the end here.
Yeah.